the characteristics of enzyme, and then we have looked at mechanism of enzyme action, which is the lock and key model, the induced fit model. So you should by now be able to explain these two models, okay, or hypotheses. And then we looked at the factors that affects enzyme activities, which are you have the substrate concentration, enzyme concentration, and then you also have the temperature and also pH. Okay, so every enzyme has their own optimum temperature and pH. Okay, to catalyze. Okay, to catalyze the reaction at maximum rate. Okay, and then after that we looked at Michaelis-Menten and also line-Weaverberg plot. Okay, so from Michaelis-Menten you have to determine the Km value of a substrate of a substrate, so that from that Km value the enzyme, okay, you know how the rate of the reaction, okay, you know the rate of the reaction from the Km value. So the substrate with the lowest Km value will have the highest affinity to bind to the enzyme. Okay. So basically Km value is the substrate concentration at half of the reaction rate, half of the velocity. And then okay, so you can determine the Km value and also the Vmax value from the hyperbolic graph, from the hyperbolic graph, but in order for you to determine the value for these two, Vmax and also Km, it is just an estimation. It is not precise because the value is really small. So the molarity is really small. So in order to get a more precise value, so you have to convert the hyperbolic graph into a straight line graph, which is the line-Weaverberg plot, where all the values for the velocity and also substrate concentration you 1 over setiap value tu. So from that you can get a straight line. Okay, so from that straight line you can determine the Km value and also the Vmax value by looking at the intercept, okay, of at y-axis and also x-axis. Okay, so please kalau rasa you are not tak faham lagi, so please do your revision lah, look back at the notes and also the video. So you have the video in the YouTube channel of acid biology, and also I have uploaded the last lecture video in my channel as well. Okay, so you can refer to those videos. Okay, so back to the notes. We are going to look at subtopic 4.5, which is on cofactors. So it says here some enzyme consists of only protein. So you know that enzymes are globular proteins, so they are made up of amino acids. So but some enzyme can only function when other chemical component is present. Okay, so this other chemical component is a non-protein part of the enzyme. Okay, non-protein part of the enzyme. Okay, non-protein part. Okay, so in order to activate the enzyme and in order for the enzyme to be able to catalyze a reaction, it needs this other component where we call it as cofactors. Okay, so cofactors is a non-protein molecule or ion needed for proper functioning of an enzyme. Okay, so the cofactor will activate the enzyme to catalyze the reaction. Okay, so this is, for example, an enzyme. So it is 100% made up of protein. Okay, so for this enzyme, we call this enzyme as apoenzyme, which is enzyme in the inactive form, without additional component, okay, without the cofactors attached to the enzyme. Okay, so apa enzyme? You have to know the definition. It is an inactive enzyme, while cofactor is the additional chemical component. So this is the cofactor. So one of the cofactor, it can be coenzyme. So cofactors, there are three different types of cofactor. You can have the coenzyme, metal ion or activator, and also you have the prosthetic group. Okay, so in this case, in this diagram, it shows you that the cofactor here is of coenzyme. Okay, so this coenzyme will bind to the, for example, to the active site of the enzyme. Okay, once this coenzyme bound to the active site of the enzyme, it causes the enzyme to be activated, and this is the combination of the enzyme and also cofactor. We call it as holoenzyme. Okay, holoenzyme, which is the active form of the enzyme. So as you can see, once the cofactor bound to the active site, so now the substrate can fit perfectly to the active site of the enzyme, and the enzyme can catalyze the reaction. Okay, so therefore holoenzyme is a complete and active enzyme. So you have to know the definition of these two. What is apoenzyme? What is holoenzyme? Okay. Okay, so cofactor modifies the chemical structure of enzyme in certain ways that enables it to function more effectively. Okay, more effectively. So three types of cofactor is coenzyme, metal ions, or we can also call it as activator, and also prosthetic groups. Okay, so cofactors can be an organic or inorganic molecule. Okay. So metal ion, for example, is an inorganic molecule. So it can be, for example, you can have magnesium or calcium ion. Okay, so those are inorganic molecule. They are ions. Okay. Okay. So the first type of cofactors is coenzyme. Please do not confuse with cofactor. Cofactor tajuk besar dia. So bawah dia ada coenzyme. Okay. So coenzyme are organic cofactors. So you have to know which one are the organic cofactor, which one are the inorganic cofactors. You have to know that. Okay. They are small organic and non-protein molecule. They will bind loosely, okay, loosely and temporarily to the active site of the enzyme. Okay, so this is how the coenzyme binds to the active site of the enzyme, loosely and temporarily. Most coenzymes are carrier molecules that transfer chemical groups, atoms or electrons from one molecule to another. For example, you have these molecules, NAD plus, okay, plus tu mesti superscript NADP plus, coenzyme A and FAD. So you will know more, okay, about these molecules when we reach to chapter cellular respiration and also photosynthesis. Okay, so these are really important molecules that we are going to look at in the next chapter. Okay, so for example, if you look at how the coenzyme works, okay, kat sini it says most coenzyme are carriers molecule that transfers, highlight that, transfers chemical group, atoms or electron from one molecule to another. So for example, here you have this molecule NAD plus, okay, NAD plus, which acts as the coenzyme for the enzyme malonate dehydrogenase. Okay, NAD plus is the coenzyme for the enzyme malonate dehydrogenase. Okay, so from this reaction, as you can see, the substrate for the malonate dehydrogenase is malate. Okay, so malate. So this is the structure of malate. And this structure there. Okay, so the reaction involves, okay, NAD plus to take the hydrogen atom from malate. Okay, so the hydrogen atom now becomes hydrogen ion. So one of the hydrogen ion will be added to the NAD plus to become NADH, and the other hydrogen ion, the hydrogen atom becomes hydrogen ion, which will be released into the surrounding environment. Okay, So malate now will be converted into oxaloacetate. Okay, so you look at the way of how the coenzyme works, kan? So it transfers chemical group from one molecule to another. So in this case, NAD plus takes hydrogen atoms from malate. It takes and add the hydrogen ion to itself to become NADH. So the reaction you see here is one of the reaction involved in cellular respiration. Okay, so cellular respiration, later we are going to look at that it has several stages. It has it consists of glycolysis, it consists of pyruvate saya lupa, pyruvate decarboxylation. Okay, process decarboxylation. And then the third one is Krebs cycle. Okay, Krebs cycle. And then the fourth one is oxidative phosphorylation. Okay, so cellular respiration, it has four stages, and this reaction, okay, is one of the reaction that involve in Krebs cycle of cellular respiration. So the detailed reaction of this we are going to look at during chapter 5, the next chapter after enzyme. Okay, so same goes to NAD plus. It has similar roles as NAD plus. Okay, but NAD plus is the cofactor of enzyme that involved for the process or the reaction involved in photosynthesis. Coenzyme dengan FAD dalam reaction yang melibatkan cellular respiration. Okay, so these are the cofactors. Coenzyme of their respective enzyme. Okay, to catalyze certain specific reaction. Okay, so the second type of cofactors are metal ions, or we can also refer it as activators. Okay, so metal ions or activators are inorganic cofactors. Okay, cofactors, they are inorganic. How do you know that a certain molecule is organic or inorganic? Belajar chemistry kan? How do you know whether that molecule is organic or inorganic? Apa dia?
Organic has carbon.
Has carbon atoms in the structure.
Yes.
Okay. So in this case of metal ion, obviously there are ions. Okay. No other elements. Okay. So there are metal ions. They also known as activators. So this is how they bind to the metal ion bind to the enzyme temporarily, secara sementara. Okay, the metal ion binds temporarily to the enzyme and changes the shape of the active site. Okay, so allowing the chemical reaction to take place. Okay, so most are trace elements that all organisms require in really small amount. Okay, so for example, magnesium ion, okay, magnesium, calcium ion, ferrum, and also zinc. Okay, so calcium ion are needed. For example, okay, this calcium ion are needed to activate thrombokinase, which is one of the enzyme involved in blood clotting mechanism, which converts prothrombin to thrombin. Okay, so this is one of the mechanism involved in blood clotting. Okay, so without calcium, tak bolehlah nak berlaku blood clotting tu secara eloknya, kan? So that is one of the example, okay, for the metal ions. Okay, so the third class of cofactors, we have the prosthetic groups. Okay, so organic molecules that bind permanently to the enzyme. So for example, we have HAM group. HAM ni awak boleh eja dia macam ni, HAEM or HEME, whichever ways. Okay, so HAME, HAM ni is the prosthetic group in hemoglobin. Okay, so it contains ferrum ion, Hem enables hemoglobin molecules to carry oxygen. So another place where you can find hem is that it is the prosthetic group found in enzyme catalase. Okay, so catalase macam kita tengok masa chapter 2, okay, which is on organelles. So catalase is the enzyme found in the organelle peroxisome, okay, where it involves in the reaction to break down hydrogen peroxide, okay, into oxygen and also water. Okay, oxygen and also water. If you were to stabilize balance the reaction macam ni lah kot. Okay. Alright. Okay, so as you can see from this diagram, okay, so the hemoglobins are pigments, okay, that are found in red blood cells. Okay, in red blood cells. So each of these red blood cells will contain several hundred millions of hemoglobin molecule to transport oxygen. So imagine this is one hemoglobin molecule in red blood cells. So red blood cell will have millions, okay, millions of hemoglobin. So each of these hemoglobin will bind to 4 molecule of oxygen because this hemoglobin has 4 subunit, okay, 4 subunit, and each of these subunit has a hem group, okay, has a hem group. So each of the hem group, at the center of the hem group will have a ferrum for the binding of oxygen, okay, for the binding of oxygen molecule. So imagine how many oxygen molecule can a red blood cell carries if it has millions of hemoglobin. So satu hemoglobin je dah boleh bind kepada 4 molecules of oxygen. Okay. Okay, so far do you have any question on cofactors? Okay, so if there's no question, so I hope you understand. So now we are going to move on to the next subtopic, which is 4.6, enzyme inhibition. So you have to know what are inhibitors. Okay, so inhibitors are certain chemicals that selectively inhibit, okay, the action of specific enzyme. So enzyme has their own substrate, specific substrate. It also has its specific inhibitors that will inhibit the reaction, okay, catalyzed by that enzyme. So the inhibitors, the way of how the inhibitors works, okay, is based on how the inhibitor binds to the enzyme. Okay, so the first type of inhibitors is grouped under reversible inhibitors. Okay, second you have the irreversible inhibitors. So why do we call it as reversible inhibitors? Because for reversible inhibitors, they will bind to the enzyme temporarily, secara sementara. Okay, the binding of the inhibitors are temporary, so it means that the effects of reversible inhibitor is only for the time that the inhibitors bind to the enzyme. But as for the irreversible inhibitors, the inhibitor will bind permanently to the enzyme and stops the enzyme from catalyzing the reaction forever. Okay, irreversible. It destroys the enzyme function. So for the first reversible inhibitors, it says it forms weak bonds such as hydrogen bonds with the enzyme. Okay, these weak bonds can easily break. Okay, enzyme revert the function two functional structures. And for reversible inhibitors, there are 2 types, which is competitive inhibitors and non-competitive inhibitors. Okay, so for why do we call it as competitive inhibitors? For competitive inhibitors, it binds to the active site of the enzyme. So for example, here you have an enzyme, the active site of the enzyme, and then you have here is the substrate, okay, for that enzyme. Okay, so for competitive inhibitors, this inhibitor has similar shape to the substrate, Okay, so instead of the substrate binding to the active site, the inhibitor will compete with the substrate to bind to the active site. So instead of the substrate binding to the active site, you have inhibitor binding to the active site and prevents the substrate from entering into the active site. So therefore, reaction cannot occur. Okay, and as for the non-competitive inhibitors, this for the non-competitive inhibitors, so for example, I drew the non-competitive as in circle shape here. Okay, so because the shape of the non-competitive inhibitor is different from the substrate, it will not compete with the substrate to bind to the active site, but instead it will bind to another site of the enzyme. We call it as the allosteric site. Okay, so for example here, okay, you have another site here. Okay, so imagine ini dah tak ada dah. Okay, so instead the non-competitive inhibitor will bind to another site of the enzyme. We call it as the allosteric site. Okay, allosteric site. Okay, so the binding of these non-competitive inhibitors will cause the shape of the enzyme to change. Okay, so when the shape of the enzyme changes, it also causes the active site to change, and once the active site changes, the substrate will no longer combine to the active site because now the shape of the active site is different from the shape of the of the substrate. Okay, so ingat reversible ada dua. Okay, competitive and also non-competitive inhibitors. For irreversible, tak boleh dah kan. So irreversible, why is it irreversible? Because this inhibitor will bind tightly and permanently to the enzyme through the formation of covalent bond. So compared to the reversible, the interaction between the inhibitors and the enzyme is through weak bonds such as hydrogen bond. But for irreversible, it forms covalent bond, a more stronger bond, so that the binding between the inhibitors and also the enzyme are strong, tight. It binds tightly and permanently, and so therefore it will destroy the enzyme function. Okay. So the first okay, so now we are going to look at the detailed explanation. Okay, for reversible inhibitors, okay, there are two types, which is competitive and non-competitive. So for reversible inhibitors, they form weak bonds. Okay, weak bonds that can easily break, such as the hydrogen bonds with the enzyme. So the interaction between the inhibitors and also the enzyme is through weak bonds that can easily break. Okay, so the inhibition only takes place whilst the inhibitor is attached to the enzyme. So the effects of reversible inhibitors are temporary. Okay, temporary, sementara saja. Okay, so for the normal reaction, okay, so you have the enzyme. So this is the allosteric site of enzyme. So this is the active site of enzyme. Okay, so the active site of the enzyme is complementary to the shape of the substrate. So the substrate binds to the active site. So this is for the normal reaction. But in the presence of competitive inhibitors, okay, so the competitive inhibitor, it has similar, not the same shape, but similar shape to the substrate. Okay, so because the inhibitor has similar shape, so it can bind to the active site, and this will prevent the actual substrate from binding to the active site. So the presence of inhibitor in the active site will prevent the substrate from binding, so reaction cannot occur. Okay, and for the non-competitive, dia tak kisah pun, it does not care about the presence of substrate at all. It just bind, okay, to the allosteric site of the enzyme. Okay, so another site of the enzyme that is not the site for the binding of substrate. Okay, so the binding of the non-competitive inhibitor will change, okay, the shape of the active site. Okay, so as you can see, initially the shape of the active site is like this. So the binding of the non-competitive inhibitor will cause the shape of the active site to change. So now the shape of the active site is not complementary to the shape of the substrate. So this prevents the substrate from entering into the active site and no reaction can occur. Okay. Okay, so sama je. Okay, the same explanation. Okay, this slide it explains even for each one, for each point. Okay, so competitive inhibitor, as I said, it has similar shape. Kalau tulis same shape, salah the shape is not the same, nampak lain kan? But they have similar shape. Okay, so that allows the inhibitor to be able to bind to the active site. Okay, so so the keyword here is similar. So ini adalah key point yang you have to include, okay, in your answer. Alright. So similar shape to the natural substrate, close to the close structural similarity. The shape is similar. The shape of the substrate is similar to the inhibitor. So because the inhibitor has similar shape, so the inhibitor will compete with the substrate for the same active site. So that's why we call it as competitive inhibitor. Inhibitor competes with the substrate to bind to the active site. So therefore, the effects of the binding of the inhibitor to the active site is that it will slow down the rate of reaction. Okay, so therefore less product produced and can be overcome by increasing substrate concentration. So this is how you overcome the effects of competitive inhibitor. You add more substrate to the reaction. So when you add more substrate to the reaction, there is a higher tendency for the substrate to bind to the active site instead of the inhibitor bind to the active site. Boleh imagine So if you add more substrate, more possibility of the substrate binding to the active site instead of the inhibitor. So this is how you overcome the effects of competitive inhibitor. Alright. So ini point yang penting Point ni penting. So yang ini juga. So soalan yang macam ni kalau selalu dia keluar pun markah dia 3 ataupun 4 markah kan. So if you have to explain on competitive inhibitors, so the first, second and the last point is really important for you to include, okay, in your explanation. Okay, so competitive inhibitors. So here you have an enzyme, okay. Okay, so an enzyme, which is the name of the enzyme is succinate dehydrogenase. Okay, so this is the blue enzyme here is succinate dehydrogenase. Okay, so the substrate is succinate. Where is succinate? Dalam gambar ni. Okay, this one here. Okay, so you have the enzyme, you have the substrate. Okay, substrate. So this is the substrate, succinate. The product should be fumarate. So the binding of succinate to the enzyme succinate dehydrogenase will catalyze a reaction that produces the product which is fumarate. Okay. But in the presence of an inhibitor which is malonate, okay, which is it is a competitive inhibitor to the succinate. So as you can see, the structure of malonate is similar to the structure of succinate. Okay, malonate has similar structure to succinate. So because of the similarity, so this enables the malonate to compete with the succinate to bind to the active site. So the binding of malonate to the active site of the enzyme produces this malonate enzyme complex and stops the reaction. Okay, stops the reaction or slow down the reaction. And if the reaction is stopped, so no product produced. If the reaction is slowed down, so less product will be produced. Okay, so ini awak bolehlah jawab dalam punya point, tapi yang paling penting yang ini. Point 1, 2 dengan 3 ni. Yang last. Okay. Okay, so as for the non-competitive inhibitors, okay, so non-competitive inhibitors, okay, they have no structural similarities with the substrate. So as you can see, the substrate, the shape is like this. The shape of the inhibitor is totally different. Okay, so the non-competitive inhibitors will bind to the allosteric site. Okay, so this is the allosteric site, another site on the enzyme which is not the active site. So binding causes enzyme to change its conformation. Okay, so you can also say here shape. Okay, the binding of the inhibitor changes the shape of the enzyme. So once the enzyme changes its shape, active site also change shape. So this prevent substrate from binding to the active site. So ditambah kat sini, from binding to the active site. Okay. So enzyme not functioning properly, slower rate of reaction and less product produced. Okay. So ini point ni sama juga dengan lebih kurang point yang untuk competitive inhibitors. Okay. So yang paling penting untuk when you are describing or explaining on non-competitive inhibitors, point number 1, no structural similarities with the substrate, bind to the allosteric site, causes the enzyme to change shape. So these are the most important point for you to include in your explanation. Okay. Yang lain tu boleh include jugalah kan to complete your answer. Okay, to complete your explanation. Okay, so allosteric inhibitors change the shape of the enzyme so it cannot bind to the substrate. Okay, so this is a graph. As you can see from this graph, it shows you it has a combination of 3 graphs actually. Okay, so if you look at the graph here, the green graph, okay, the green line here, it shows you a normal reaction without the presence of any inhibitors. The green graph, okay, uninhibited. Okay, the green graph shows normal reaction. Okay, normal reaction without the presence of any inhibitor. So as you can see, the reaction is really high, it occurs really fast and reaches the maximum velocity quickly. Okay, but if you add, okay, in the reaction you add competitive inhibitor to the reaction, as you can see, the rate of reaction will slow down. Okay, will slow down and produce less product, but eventually it reaches the maximum velocity, maximum rate of reaction if you keep on increasing the substrate concentration. Tadi kan, how do you overcome competitive inhibitor? By adding more substrate. Okay, or you increase the substrate concentration. Okay, adding more substrate or increase substrate concentration. But for the red graph here, okay, as you can see, the reaction rate slows down even more and it stops at certain point. Okay, at a certain point it stops. Okay, like for example here, okay, sampai sini dia akan stop. So it shows you for the non-competitive inhibitor, because it binds to the allosteric site of the enzyme, dia tak kisah pun pasal active site. So there is no use, okay, of you adding more substrate. Okay, so adding more substrate to the reaction in the presence of non-competitive inhibitor will not work. The reactions will remain slow and stop at certain point. Okay. So increasing the substrate concentration does not affect the rate of reaction. So you have to know the difference between graph 1, graph 2, and also graph 3. Okay, so you have to know which reaction is the normal reaction, which reaction occurs in the presence of competitive inhibitors, which reaction occurs in the presence of non-competitive inhibitors. Okay, so the next type of inhibitors is irreversible inhibitors. The effects is permanent. It destroys the enzyme permanently. Okay, so irreversible inhibitors or non-reversible inhibitors will bind tightly and permanently to the enzyme with strong covalent bonds and destroys the enzyme function. So this can occur at low inhibitor concentration. Okay. Okay, so for example, okay, toxin and poison. Okay, toxin and poison can act as irreversible inhibitors. It destroys the enzyme function permanently. For example, sarin, which is a type of a nerve gas. Okay, a toxin. Okay, toxin, which is a nerve gas. So it has no odor, the bau, no taste, and it targets the nervous system. Okay, so the serine will bind covalently to the R group of this neurotransmitter. This is a type of neurotransmitter, acetylcholinesterase. Sorry, bukan neurotransmitter, the enzyme for a neurotransmitter. Okay, acetylcholinesterase. Okay, so this is an enzyme. Okay, an enzyme for the neurotransmitter acetylcholine. Okay, acetylcholinesterase is an enzyme for the neurotransmitter acetylcholine. Acetylcholine, okay, is a type of neurotransmitter that allows for the transmission of impulse between between neuron and muscle cell. Okay, so the transmission of impulse from neuron to muscle cell causes the muscle cell to be able to contract. Okay, so you do not want this acetylcholine to remain binding to the muscle cell, as it will cause continuous transmission of impulse to the muscle cell and causes the muscle cell to continuously contract. Kalau tak nak muscle awak tu sentiasa contract, dia akan menyebabkan kepada kejang. Okay, so this acetylcholine has to break down by this enzyme, acetylcholinesterase. Okay, acetylcholine will be broken down by this enzyme. But in the presence of serine, okay, it will stop or prevent the reaction to break down acetylcholine. Okay, so kita tengok ni. Serine binds covalently to the R group of acetylcholinesterase, altering the shape of the active site. So acetylcholine, which is a neurotransmitter, cannot be broken down. Acetylcholine molecule accumulates in the synaptic cleft and causes continuous contraction of nerve impulses, leading to convulsion, paresthesis, and eventually death. Okay, so acetylcholine is a type of neurotransmitter that causes transmission of impulse to occur between neuron and muscle. Okay, so you do not want the transmission of the impulse to occur continuously, as it will cause convulsion, paresthesis, and eventually death. Continuous contraction. Okay, continuous contraction. Okay, you do not want that. So you have to prevent the continuous transmission of impulse by breaking down the acetylcholine by the enzyme acetylcholinesterase. So the serine akan menyebabkan acetylcholine cannot be broken down and causes continuous transmission of impulse. Okay, ni sepatutnya continuous transmission ni. Okay, transmission of nerve impulses. Okay, faham ke? Do you understand? Okay. Okay, so the next slide. Okay, the next subtopic. So, wait. Okay, so I hope you understand on the differences between reversible inhibitors and irreversible inhibitors. Okay, reversible, we have two types, which is competitive and non-competitive. All right. Okay, so if you are okay with that subtopic, so now we are going to move on to the next subtopic, which is 4.7, which is regulation of enzyme activity when reaction occurs and at what rate. Okay, so it says here metabolic pathway in a cell is regulated by activity of enzyme. So your cell, okay, every cell in your body is really active. Every cell is involved in many different types of reaction that is catalyzed by enzymes. Okay, by many enzymes. Okay, so what regulate the activity of this enzyme? Okay, there are certain mechanism that regulate the activity of enzyme within cell. For example, okay, so number one here. So dibagi banyak example here. Okay, so it says here genes that code for enzyme may be turned on or off. Okay, for example, gene that codes for beta-galactosidase is activated when lactose is present. Okay, in this case, the enzyme is beta-galactosidase. Beta-galactosidase is an enzyme that is found, for example, in a bacteria E. coli. Okay, E. coli. So you have a bacteria, the E. coli bacteria, and then you grow the bacteria in a medium containing lactose. Okay, in a medium containing lactose. So in this bacteria, it has its DNA molecule. Okay, it has its DNA molecule. So the DNA molecule within the E. coli, there is a segment of the DNA, a gene, okay, a gene of the DNA that is used to code for the synthesis of the enzyme beta-galactosidase. Ini adalah region pada DNA tu yang akan code for the synthesis of enzyme beta-galactosidase. Okay, this gene can only be turned on. Okay, turn on tu maksudnya kod tu boleh digunakan untuk menghasilkan enzim tu. Turn on, it can be turned on in the presence of lactose. Okay, so the presence of lactose in the medium that the bacteria is grown has lactose. Okay, the presence of lactose will cause the gene to be turned on and the enzyme beta-galactosidase can be synthesized. Okay, but without the presence of the lactose, the gene will be turned off, okay, not activated, and so therefore the enzyme beta-galactosidase cannot be synthesized. Okay, itu maksud dia. So the synthesis, so it applies to all, to all situations. So in order for a certain enzyme to be synthesized by a cell, mesti ada faktor yang menyebabkan enzim tu boleh dihasilkan oleh sel tu. Okay, dengan mengaktifkan gene untuk menghasilkan enzim tersebut. Faham Boleh So the second situation, it says here inactive enzyme. Okay, such as for example pepsinogen. Your stomach, okay, contains your stomach contains HCl, hydrochloric enzyme, hydrochloride, HCl kan. Why does the stomach needs to produce HCl? The presence of this HCl, okay, will activate the enzyme pepsinogen into pepsin. Okay, initially before pepsin is produced, it is in the inactive form. So the inactive form is pepsinogen. In order to activate the enzyme pepsinogen, has to have an acidic condition, which is HCl. So the presence of acidic condition will cause the pepsinogen to be activated into pepsin. So once the pepsin is activated, it can be used to hydrolyze protein. Okay, so samalah apply kepada enzim-enzim yang lain kan. So inactive enzyme has to be activated by certain factors. Okay. So the third one, presence of inhibitors. Okay, presence of inhibitors such as reversible inhibition. Macam tadi lah kita tengok kan. So the presence of inhibitor will inhibit or slow down the rate of reaction catalyzed by the enzyme. Okay, so the presence of inhibitor will slow down or stop the reaction. So that will also regulate the enzyme activity. Okay, the fourth one, regulator molecules may activate or activate or inhibit enzymes such as allosteric regulators. Okay, so there are molecules in cell that is to function to either activate or inhibit the enzyme. Okay, so these are what we call it as regulator molecule or allosteric regulators, because the molecule that function as activator or inhibitor will bind to the allosteric site and either activate or inhibit the enzyme. Okay, so most enzyme initially is in inactive form. Okay, it is in active form. You have to activate the enzyme. Okay, so in order to activate the enzyme, you need activator. But in order to inhibit the enzyme, you have to have inhibitor. Okay, so this molecule, we call it as regulatory molecule that binds to the allosteric site. So therefore, we can also call it as allosteric regulators. The last one is end products. The end product produced by an enzyme can also be the factor that influence the enzyme to catalyze the reaction. The end product. Okay, so end product may inhibit enzyme. So this is what we call it as feedback inhibition. Okay, so you have the first enzyme, for example, first enzyme catalyze a reaction. Okay, sorry, bukan macam tu. So you have this first substrate. Okay, first substrate catalyze first substrate involved in a reaction catalyzed by an enzyme. Okay, by an enzyme. So this is the enzyme. Okay, so produce the second product. Second product will be converted into a third product and converted into another product. Okay, so the last product here, the star yang saya wakilkan dengan star ni. So this is the end product. Okay, so you have a series of reaction catalyzed by many enzyme. So enzyme 1, enzyme 2, enzyme 3, for example, until you get the end product. Okay, so this end product can act as inhibitor, okay, for the first enzyme of the reaction. Okay, for the first enzyme of the reaction, usually the enzyme that involved in the first step of the reaction. Okay, so this is what we call it as the feedback inhibition. The end product may become inhibitor for the first enzyme of the reaction. Okay, so these are the example of how enzyme activities are regulated in the cell. Okay, so dia bukan sesenang. Okay, you have substrate, you have enzyme. So the substrate binds to the enzyme, and the enzyme catalyzes the reaction to produce product. It's not as easy as that. So the activity of enzyme are regulated by many conditions. Okay, by many conditions and also by many factors. Okay, within the cell. Okay, so far, do you have any questions? Faham daripada awal? Okay, so if you do not have any question, I guess we stop up to this slide. Okay, so we are going to continue after the holiday. So next week you have your test. Okay, and then after that you have your